Hello. Okay, good. Hi, everyone. We'll just let people file in and make themselves comfortable. Don't hang out too much at the back. Don't be shy. Come on up to the front, students. Come on. Everybody can come up and occupy the seats that are here. So I just want to welcome everyone. Good afternoon to faculty, students, postdoctoral fellows, research associates, and staff of the Department of Physics. And good afternoon to those of you in the broader University of Toronto community. Through our streaming feeds. Today, I'd like all of you to consider yourselves members of the Department of Physics at the University of Toronto. My name is Paul Kushner, and I have the privilege of serving as chair of this fantastic department. And in that role, I'd like to extend a very warm welcome to the 2026 Welsh Lectures in Physics. Now, out of curiosity, how many of you are attending the Welsh Lectures for the first time? Wow, look at that. That's wonderful to see all of you here. And I hope that all of you online also raised your hands in support. So it's great. We have about at least a third of the people here who are attending for the first time. So it's an especially warm welcome to you newcomers. And we hope to see you back next year. So as I extend words of welcome to you, I'd also like to share with you the official U of T statement of acknowledgement of traditional land. I wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. Today, this meeting place is still the home to many Indigenous peoples from across Turtle Island, and we're grateful to have the opportunity to work on this land. So since 1975, we've been holding the Welsh Lectures in celebration of the legacy of the late Professor Harry Welsh, who lived from 1910 to 1984. Each year, we invite two physicists at the leading edge of the discipline to share their expert perspectives on cutting-edge topics. So today you'll hear, if you bear with me while I flip my notes and juggle this mic. So today you'll hear about the tremendous work framed at a high level and intended for a broad public audience. Tomorrow, there'll be two colloquia, and the series will continue with these more specialized talks that focus on topics of their current research. But everyone here is welcome to attend, and maybe by the little bit of training you had today, you'll be prepared to hear a bit more about these topics in depth tomorrow. So you're going to hear more about our speakers shortly, but what about Professor Welsh? He was a fellow of the Royal Society, a distinguished molecular spectroscopist, a child of the department in his undergraduate years, a professor in the department starting in the 1940s, and chair of the department in the 1960s. I'd say that in important respects, this is Harry Welsh's department. He presided over the department through its move in the 1960s into the newly constructed McLennan Physical Laboratories, which we still occupy today, not quite as new, and established our modern department through disciplinary broadening and tremendous growth. As an aside, and speaking of growth in our department, I'd like to take this chance to introduce a new colleague who has joined the department's faculty since the last Welsh Lectures last year. Indeed, it's great to share with you that Dr. Valentin Crepel, who is way in the back there, you can see him like in the back corner, like a good undergraduate student hanging at the back of the class. It's great. Joined our theoretical quantum condensed matter group this year. His research is focused on the theory of so-called moiré materials, layered quasi-two-dimensional crystalline systems, which have over the last decade revolutionized condensed matter physics. Yeah, and you might remember that last year we heard about these materials from an experimentalist perspective during Pablo Herrero's lectures. So Dr. Crepel is expanding theoretical research in this field, discovering new states of matter in these systems. So a warm welcome of our new colleague to Harry Welsh's department. It's a profession. Welcome, Valentin. I'm sure Harry would have appreciated your research. So this year, it's a great honor for us to host two wonderful guests whom the session chairs will be introducing shortly. Professor Stephen Quake is the Lee Otterson Professor of Bioengineering and Applied Physics at Stanford University, who combines physics, biology, and biotechnology to drive new ways of measuring biological systems. Using this, he learns about how they work and how this knowledge can drive important medical applications. So we'll be hearing from him shortly. And then Professor Tony Noble of the Department of Physics at Queen's University, he holds there the Canada Research Chair in Particle Astrophysics and serves as the scientific director of the Arthur McDonald Institute and is therefore playing a critical role in leading the increasingly intensive search for dark matter out of the Snow Lab facility. So you'll be hearing about him and from him shortly. So before we continue and get to the lectures, I just wanted to thank the 2026 Welsh Lectures Committee. This is chaired by Young Beck Kim, who's sitting there in front, and the staff who have worked so hard behind the scenes and on the scene to organize the lectures this year, which also includes those who've organized stuff in the lobby and over in physics, as well as our AV workers here. So for now, I'll say thank you to Welsh Chair Young Beck Kim, and let him thank the rest of the Welsh team in detail following the talks. Finally, a reception will follow the lectures around 4:30 p.m. in the McLennan Physics Undergrad Lounge. That's MP 125. I don't expect you to find it on your own. Just follow the crowds. Many of our department members know exactly where to go. So I welcome everyone to join us there. There's room for all, and there'll be snacks for all as well. So for now, I'd like to invite Professor Sid Goyle to introduce our first speaker. Sid, go ahead.
Thank you, Paul. My name is Sid Goyle. I'm a faculty here in physics, and I work at the interface of physics and biology. And I'm delighted to introduce Professor Stephen Quake, who is our first Welsh speaker. Steve is, as Paul said, Lee Otteson Professor of Bioengineering and Professor of Applied Physics at Stanford, and was the president of Chan Zuckerberg BioHub for the last 10 years, shaping their effort in single-cell biology, and notably building a fantastic team to address some of the fundamental questions, which I'm sure will continue on. Professor Quake is an interdisciplinary scientist and an entrepreneur, which is exemplified by being one of only 24 scientists elected for all the 3 national academies, which I imagine is engineering, medicine, and science. But his roots are theoretical physics. Professor Quake earned his BS in physics and MS in mathematics from Stanford, then did a PhD in theoretical physics at Oxford University. His thesis was in statistical mechanics and the effects of knots in polymers. He's going to talk about more polymers today, I guess. I have heard from the grapevine that he considered getting 2 PhDs, one in theory and one in experiment. He can confirm if that's true or false. With that, please join me in inviting Professor Steve Quake. Thanks.
Thank you. It's terrific to be here in Toronto. It's been a terrific day. I'm looking forward to the rest of the visit. It's been said that physics is the study of the very small, the very large, and the very complex. And this lecture will be about the very complex, and that is to say about life. The fundamental, sorry, Zoom is being a little too helpful. Let me try to make that go away. Let's try that now. Okay. All right, almost. There we go, much better. Sorry for that. So cells are the fundamental quanta of biology. They're really the smallest indivisible unit of life. This is a painting by a scientist and artist named David Goodell, where he's envisioning what the last universal common ancestor of life would be, the first cell, so to speak. And he's made a beautiful series of paintings where he's inspired by the actual structures of molecules, in this case, the crystal structures of various proteins, and tries to put them into context. And so you can see here, he's got a cell that's dividing into 2. The cells, you can see the interiors are very dense. These are bacterial cells, so there's no nucleus, but there's many different protein machines there that are doing different things. There's the cell membranes, sort of lipids and sugars on the surface. And there's this long string thing, which is the genome. That's the DNA. I'm going to talk both today and tomorrow about how, to what extent you can think about DNA as the blueprint of the cell or of the organism, and whether that's a good metaphor or not, and what is actually useful for and what it's doing and what parts are missing from that kind of picture. So let's start by talking about what DNA is. It is sort of the genome. It's the molecule of genetic inheritance. Its structure was famously solved to be a double helix here. So the information is encoded with a chemical alphabet. There's 4 different chemicals, adenine, thymine, guanine, cytosine, that pair up in the manner shown here. And so there's a redundancy to the code, and it's as if you had a ladder and just twisted it around. And the inner rungs are where the information is stored. And that information is what encodes the sequences to make proteins and other molecules within the cell. And the discovery of this double helical structure very famously recounted. The 3 principles were James Watson, Francis Crick, and Rosalind Franklin. And sort of Crick and Watson went around saying they had discovered the secret of life when they solved the structure of DNA. That's probably a bit of an overstatement, but they made a lot of hay out of it. And you can read about these discoveries in their own words in the cases of Watson and Crick in these 2 books. And Franklin tragically died very young, but there's a wonderful biography here by Brendan Maddox, which kind of lays out that kind of moment in science. Since we're in a physics crew, let's talk about DNA by the numbers here for a moment. Every cell in your body has 2 meters of DNA. DNA is a polymer, it's stringy like you saw in the painting. The actual proportions, every cell in your body, you've got the 23 chromosomes, 2 copies of each, but if you laid them all end to end, that'd be 2 meters long, a molecule or set of molecules 2 meters long. And you have roughly 30 trillion cells in your body. And so if you were to take all the DNA from all the cells in your body and lay it end to end to end, it would go all the way to the Earth and the sun and back more than 200 times. How does that work? Well, it works because DNA is macroscopic in one dimension, but atomic in the other 2. It's got a diameter of only 2 nanometers. And so it actually can be compressed into these very small volumes and spaces. And I'm going to show you a movie here I made. Sid mentioned I had done my PhD work and trying to understand this intersection, something about the intersection of topology and polymers. And that was a theoretical work. But I then spent a decade as experimentalist trying to actually tie knots into DNA to study the statistical mechanical properties. And we finally did it. And this is a movie showing that. And so I'll start the movie in a minute, but I want to just explain what you're going to see. So it's a very long DNA molecule, maybe 30 microns long, anchored to 2 beads, 2 micron-sized beads on the end. And you're able to see the molecule because it's fluorescing. This is something that ties into Harry Welsh's legacy, right? He was famous for doing Raman spectroscopy, where you shine in light at one wavelength, the molecule absorbs it, energy changes, it emits wavelength of a different energy, different wavelength. Fluorescence is sort of like that. You excite the molecule one wavelength, there's energy lost due to internal electronic motions that emits wavelength of another light of another wavelength, and you can have very sensitive detection. So this is basically that. And now let me show you what happens. And the ends of the beads are held in optical traps. So these are for the AMO people, like a dipole trap, focused laser beam that traps the particles and thereby lets us manipulate the molecules by manipulating the beams. Okay, here it goes. So here we're moving the stage, but then adjusting the traps to go up and down and wrap the molecule around itself. So having made a loop, we can make a very complicated knot. And you see there's a second molecule whose end is untethered there, also attached to that bead. And when the knot is done, we're going to stretch it out. And you can see that second molecule is going to wiggle its way out because, of course, Brownian motion is driving everything here. And it turns out when you tie a knot in a molecule as opposed to your fishing line, it actually tries to wiggle itself out through something called self-reptation, essentially the thermal motions. So this is the physical instantiation of the genome. And this was something that fascinated me for a long time at the beginning of my career. I was very interested in using DNA, not for the biological information it's stored, but for its physical behavior as a polymer. But of course, the whole world of biology is interested in this other aspect of information storage. And we are at kind of the tail end of an amazing revolution in instruments that can read out the information in the DNA molecule, that 4-letter alphabet that I referred to earlier. And the cost of sequencing DNA has gone down exponentially. And I was involved in this a little bit, and we developed the first single molecule sequencer. But I think the take home is that nearly all sequencers these days, the vast majority of sequence generated is based on using biophysical principles of fluorescence, like you saw in that movie. And we've, just generate an enormous amount. And it's become routine to sequence entire genomes and also to use sequencers to read out other aspects of information within the cell, which I'll get to in a moment. So let's talk about how that information flows. And this term central dogma was invented by Brenner and Crick to try to understand. Crick wrote an essay where they're trying to sort of formulate how does information flow in this process of synthesizing proteins. Proteins are the machines in the cell that do all the work. Their sort of blueprints are stored in the DNA. And the way that information flows is there's an enzyme, one of the protein machines, which will copy the information from DNA into RNA. That's called transcription. And then there's another protein machine called the ribosome, which will translate that by using the RNA as a template to make the protein. And that has been called the central dogma. And when a cell divides, the genome gets copied, and so that gets sent to the next generation, and that's replication, so it's DNA going to DNA. And some viruses can copy from RNA to DNA through a process called reverse transcription. And this is something that's really a universal process of biology. Jacques Monod sort of overstated a little bit by saying anything true of E. coli must also be true of elephant. E. coli is the bacterium in which most of this was discovered as sort of the hydrogen atom of biology. But it's not too much of an overstatement. The spirit, there's something there. There are these really universal principles that all organisms share. And the story of all these things, information flow, solving the structures of proteins, solving the structure of DNA, is beautifully told in this book, The Eighth Day of Creation by Horace Freeland Judson. And so now let's turn to this idea of the blueprint. It's really, for multicellular organisms, not accurate to say that the genome is the blueprint, because essentially every cell in your body has the same genome in it. And yet those cells become very different in their identities. A neuron is very different than a heart cell, very different than a lung cell. And they all have the same genome. So how does that happen? We're going to talk about that during the course of this talk. I think the proper metaphor is that the genome is a parts list, and different cell types pick and choose among these parts to create different identities and functions. And so I think the right metaphor to have is a parts list. And Sidney Brenner, great biologist, articulated this 25 years ago as an open problem. The genomes had just been sequenced, and he was trying to poke the community and say, the problem is not solved yet. Having the genome leaves you with many open questions. And he, in his pithy way, sort of summarized it by saying, biology is drowning in a sea of data and starving for knowledge. And I feel like those words really ring true today in the AI revolution, where everyone thinks it's all about getting as much data as you can. And I like to tell people, don't conflate data with knowledge. They're very different things. And this is a good example of that. Okay, so one of the things I've been obsessed with for the last decade has been trying to understand how many cell types arise from one genome. That's a restatement of the question I just articulated. Do you think of the genome as a sort of biological computer code which controls cells and development in a completely deterministic fashion? Or is it how does temperature and fluctuations come in? Because these are all single molecule problems, and so these fluctuations matter. And there's many, many open problems in interpreting the information content of the genome. The one that I'll just focus on today and tomorrow is you cannot predict the cell types of an organism from the genome alone. So if you were discovered a new species running around in the wilds of Africa somewhere that never been seen before, and you took a blood sample and you sequenced the genome of that creature, you would not be able to predict what the cell types were. That seems like a very fundamental hole in our knowledge in biology. And so we've sought to go after this problem experimentally for the moment. And so here's a thought experiment. What if you could take a cell and enumerate every molecule inside that cell? Just make a list, an inventory. Forget where they're located, forget the structures they're forming, whether in the nucleus or elsewhere. Let's just make a list of all the proteins and metabolites and such forth in a cell. Would that provide a useful estimate of cell state and cell identity, that sort of coarse graining? I think most people would agree that would be a very useful thing to have. It's not experimentally tractable to do it, but you can do a subset of that. You can count every RNA molecule in the cell. And I'm going to focus for the purposes of this talk on messenger RNA, which are the ones that are being transcribed, that intermediary between gene and protein that I showed in an earlier slide. So those messenger RNA molecules, it is possible to measure them from single cells by counting the molecules, and they provide you with a list of which genes are turned on in that particular cell. And I hope to convince you by the end of the talks today and tomorrow that we're actually discovering a lot of useful biology from that. Now, how do we do that measurement? Through a method called single-cell genomics, single-cell transcriptomics. And it's very popular right now in biology. It's one of these sort of white-hot things, and it's sort of a trendy thing of the moment. And I like to remind people that this was actually 30 years in the making. And there's an old vaudeville joke that it takes 20 years to become an overnight success. It's sort of like that in science in a lot of areas too. And so it dates back to actually the late 1980s. So in the late 1980s, Kary Mullis invented the first biochemical amplifier. So as physicists, you're familiar with electronic amplifiers and transistors and ways to do that in solid state. There are ways to amplify biological molecules as well, particularly DNA, through a process called polymerase chain reaction. It provides exponential growth. And it's become a very powerful, well, everyone knows about PCR, I guess, because of COVID tests, right? Yeah, that's where that comes from, it's the biochemical amplifier. So, and that was to amplify DNA. And almost from the moment that discovery was made, people were creating variations of it and trying to find other ways, other versions of the amplifier. And in 1990, two different folks, Jim Eberwein and James Isco, separately published papers showing a way to amplify the mRNA from a single cell using a method like PCR. So they could make an amplification of all the mRNA in a single cell. Problem was they didn't know how to read it out. There was no way to read out sort of that kind of complex chemical mixture. So they were using something called a Southern blot to look at one or two genes and show that it kind of worked. And then a little more than a decade later, 4 different groups published papers showing how to use what are called hybridization microarrays to read out the complexity. Then it went to sequencing, and that's sort of the method of choice. Now that high throughput sequencing has matured, we just sequence all those amplified molecules, and it provides a very powerful way to count them. So up to this point, all the basic building blocks were in place to do this single cell transcriptomics. And the only thing that was missing was scale. These were all sort of bespoke experiments done on half a dozen cells. Everything was very laborious and done sort of by hand. And this is where I got involved with the field. My group had been researching what's called microfluidics for many years, building miniaturized plumbing as a way to automate biology. And we had figured out how to build chips that had miniature valves on them, tens of thousands of valves, and we thought of them as the analog of the integrated circuit. And it was super fun to explore, both as a tool for biological automation, but also as a tool to understand fluid mechanics at small length scales. It turns out that for whatever reason, the laws of physics work out so that as you scale length scale, you get to very interesting regimes. The dimensionless numbers change much more quickly than they do, for example, in scaling temperature. When quantum takes over for classical mechanics, you got to get to very low temperatures. That happens much more quickly with length scale and fluid mechanics. And so things like Reynolds number, Grashof number, Peclet number, capillary, all that stuff becomes very accessible at the tens of micron scale with behavior very different than what we're familiar with at the macroscopic scale. And that was an enormous part of the fun of the research. And so these chips ultimately were used to build automation, to automate the single cell genomics process and do it on hundreds and thousands of cells at a time, first with valves, then with two-phase flow. And we've sort of reached the point where this is now routine. The devices are commercially available, and it's had an enormous impact on the field. So the way the experiments work is you take your tissue that you're interested in, you cut it out, you chemically digest it so that the single cells are separated in solution. You use one of these microfluidic devices to separate the cells, amplify the cDNA, the RNA, turn it into DNA, and you barcode every molecule so you know what cell it's from. Then you put it on a sequencer where you can count them so you know which cell and how many molecules of each species were there. And then you get to compute on it. And this is kind of a really fun aspect of it. There are roughly 20,000 genes in the human genome. So every cell is described by a 20,000 dimensional vector. Each dimension of that vector represents a different gene. The number in that component tells you how many mRNA molecules of that gene were expressed in that cell. And so it's a very data-rich way to characterize these cells. And what you can do is you can cluster them together in the 20,000 dimensional space and see if it makes biological sense. And in these pictures, which maybe you've seen in talks recently, every dot there is a different single cell. It's a 20,000 dimensional vector that's been now projected down into a two-dimensional space so you can see what the relationships look like. And the colors represent differences found in the clustering in the high-dimensional space. And there's a lot of sophistication and thought that goes into how you do these sorts of computation analyses. And we'll talk a lot about that tomorrow. So I was motivated to try to take these technologies and apply them to whole organisms. And we did that. That was kind of the reason why I got involved in the Chan Zuckerberg Philanthropy and started their first research institute, was to assemble a large team to do a collaborative project on this. And we did this for mouse and fly and lemur and human. And this was sort of a very different kind of science than I'd done before, where I was just always working in my lab. These were 24 groups. The papers have 150 authors on them, and it required a kind of coordination and teamwork that was quite different than anything I did before. And it was a ton of fun to do. And I'll tell you some of the work that came out of that. If you're interested in the history, I wrote an essay about that a couple of years ago called "A Decade of Molecular Cell Atlases," which summarizes a lot of kind of not just the work that I was involved in, but others in the community, and kind of puts that into context. So let's jump into a little bit of the science. We'll start with the fly just for a moment, because I'm going to come back to it a bit later. The fly is one of the great model organisms of biology, and there's a huge community of people who work with it. They approached us to try to build an atlas of the fly. And we did that, 17 different tissues, found more than 250 cell types. It turns out you can dissect the flies. The folks in the community have gotten very good at it. Even though they're very small, you can cut out different tissues. And so we had different tissue experts, each of whom focused on a different tissue. They would dissect out the tissue from the flies. We would then dissociate the cells, analyze them, and so forth, and put together this whole atlas. And here are two different parts of the atlas, the body wall and the testes. And you can see that there's a lot of complexity in terms of the different cell types. The human experts went through and annotated every cell type, gave it a name, based on the genes that were expressed there. And many of these cell types were sort of known before, but only one gene was used to identify them. Now there's thousands. And that represents the real advance in the power of the cell atlas. These cell types, which were previously defined by only a single gene or a couple, now have a full molecular definition. It's been very, very powerful. And I'll come back to a specific example of that later. Talk about mouse for a minute. This was in terms of chronological time, this was the first one we did. The fly came later. And this came out of a collaboration with Tony Whiskere, shown here, who's a good friend and colleague at Stanford, who was interested in looking at aging mice and what happened as they got older, and the biological effects of aging. And he was able to have mice in his lab that were 30 months old, which turns out to be the equivalent of a centenarian in human years. So these were like the equivalent of human centenarians. And he wanted to look at gene expression in them, and it come to me for help. And I said, we've just started this institute. Let's do a single cell atlas of all these mice as they age. The problem was that not many mice survived to 30 months. And so we realized we were going to have to make the best use out of each mouse that we could. And so this was the beginning of sort of the teamwork, where we had upwards of 18 different tissues from each individual mouse. And every morning in that summer, this group of students and postdocs would show up at Tony's lab, as Nick at the left would sacrifice one of the mice. They all had ice buckets. They take their tissue back to their lab, do the dissociation, the processing, bring it to the biohub where we sequenced it. And we did this through many different mice at different ages, and put together the first whole organism mouse atlas. And this is an example of one time point, the first time point we did. And it's sort of interesting because it comes back to what information is there in that 20,000 dimensional vector. So here we asked the computer to cluster all those vectors together, and then projected it into the two-dimensional space, mapped it. They're colored here by which tissue they come from. And you can see that the tissues are being resolved by that 20,000 dimensional vectors. And the cell types have all been labeled with a text there by what the human annotators did after clustering. And so what's sort of amazing is you can see that most of the clusters are a single color, meaning the tissues are resolved. Some of the clusters are multiple colors, and those cell types are cell types that are common across more than one tissue. So B cells up there in the middle. Sorry, I've got a pointer. B cells right there, multiple colors, because you have those immune cells in many different tissues. Endothelial cells also found in many different tissues. And so this was very encouraging to us because it showed that we weren't getting ourselves into trouble by having sort of systematic errors in the different tissue processing. The cells are still similar enough that the computer recognized them as being the same cell type, even though they came from different tissues. So let's talk for a minute about why we're doing this at a single cell level and not just grinding up the whole tissue and doing a bulk measurement. And the analogy that people in the field like to use is like making a fruit smoothie. You can, if you're interested in fruit, you can take it and grind it all up, make a smoothie, drink it, do chemical analysis, see what you like, try to reverse engineer what's in there. But it's hard to tell the difference between, let's say, the strawberries and the blueberries. Whereas if you were to separate all the individual fruits ahead of time and analyze the strawberries separately from the blueberries, from the raspberries, you could then have a much better understanding of the constituents and what was correlated within each fruit and what wasn't. And this is exactly what comes into play when you're trying to understand something like aging. And if you ask, what gene expression is changing with age? If you're to do this only on bulk tissue, you would not be able to discriminate between let's say you made a measurement on bulk tissue and you saw that a certain gene changes expression with age, you wouldn't know whether that came because every cell expressing that gene was changing its expression level, or that more or fewer cells were changing expression of that gene and keeping it the same. Is that sort of clear? There are two possibilities, and with the bulk measurement, you can't tell them apart. With single cell, you can. And we decided to look at how often each of those things matter. Is it more cells expressing the gene, or fewer cells, or is it every cell changing its level of expression? So, we looked at that across our mouse aging atlas, and this axis here is sort of number of cells expressing the gene. This is the intensity of the expression, and you can see for many genes whose expression changed, both things are happening. It's not one or the other. It's often both things happening, and so it really helps to be able to separate them out. And here's an example of that in the liver. You can see, for example, here's genes whose expression is going up with age, down with age. Here's some analog manifestations of that. These are the ages of the mice right here. And here's sort of number of cells of a given cell type. And so this is not even expressing, this is changes in gross numbers of cell types over age. And all these things are confounded together in the bulk measurement, and we're able to tease them apart in the mouse and learn a ton about what's happening with natural aging, including many other aspects for those who are biologists in the audience about the immune system. We could look at clonality of B cells and T cells. We could look at mutational load with age and see how that was changing in different cell types. And it was just sort of, it's been a great and very useful reference atlas. We even decided to look at what happened when you reverse aging. And so Tony and his few colleagues have been interested in this phenomenon called parabiosis, which is Latin for living together. And it turns out what you can do is sort of a Frankenstein experiment where you take two mice and you suture together their vascular system. So they're sharing blood with each other. That's called parabiosis. And what the aging community has begun to do is to parabiose together an old mouse and a young mouse. Okay, that's the middle one here. That's called right here, heterochronic, isochronic if they're both old or both young, heterochronic if they're different ages. And in this case, what they had observed was the old mouse seems to become rejuvenated. By certain measures of physiology, it's like that young blood is making that old mouse act younger. And similarly, the young mouse suffers accelerated aging. And so we decided to build an atlas of these mice. And, I thought we were going to disprove the whole thing. I thought it must be wrong. It turns out to be very subtle and interesting. And I'm really glad we did it. And you can read the paper. I'll just sort of summarize here by showing what happens in sort of the power of having the atlas by looking at all the cell types at once. We looked at one of the most sort of uniform effects of aging that changed in parabiosis. And it's genes that are involved in electron transport, those that are involved in making energy for the cells. And this is something that gets reversed on parabiosis, and it happens across so many cell types. It's not specific to particular tissue, and it's really one of the most important universal aspects of aging, and it just sort of jumped right out at us from these experiments. Okay, let's talk about human. So we went on then, having practiced on mouse and fly and lemur, which I won't talk about today, to try to build a human cell atlas. And we took the same sort of team-based approach of having tissue experts, trying to get as many tissues as we could from the same donor. And you see many of the key people involved. I'll just highlight Bob Jones on the left, who quarterbacked the whole thing. And this is sort of the state of what we're calling the Tabula Sapiens. Here, each column is a different donor, and I'll talk about them in a moment. Each row is a different tissue. Each circle is the number of cells we got from that tissue, from that donor. Things to the left of the dotted line were published in our first paper a few years ago. We've gone back and now have several other donors where we really increased our ability to get many tissues from the same individual donor. And we thought that was going to be important in this case because we wanted to be able to control against genomic differences, epigenetic differences, differences in exposure, age, and whatnot. And by getting as many tissues from the same donor, we could then be able to have a better basis for comparing them. And so we've done that. Same process with mouse. Experts annotated it. And we have, these are together some of the largest human annotated data sets in the field, which make them incredibly useful for benchmarking various algorithmic approaches. I'll just say our partner in doing this was an organization called Donor Network West. They're a nonprofit organ procurement organization. They're the ones who help people who need a transplanted organ in Northern California and Nevada. And tissues that aren't transplanted can often be used for research. And they helped us work out how to do this with a number of donors. And so, and it was sort of by far the most complex thing they'd ever undertaken as a research project. They were terrific. They have been terrific. And none of this would have been possible without them. We would get a call from them. They said, we have a potential donor who's, sort of motorcycle accident, they're brain dead. We're going to try to transplant the organs in another day or two. Does this look like someone suitable for your study? And we would look at the file, and if it looked suitable, they would then try to consent the family for the research. And this seemed to always happen in the middle of the night, that they would do these the transplant surgeries. And these were, just sort of heroic experiments that required 24 to 36 hours of nonstop work. The surgeons would be in there, they'd take out the organs to be transplanted, then one by one, take out the ones for research. They'd be ferried in ice buckets to the labs, dissociated, handled. And this sort of shows you some of the really dedicated students and staff who were and postdocs who were doing this work. And this is sort of the scale of a single experiment. And you can see a lot of this was done in COVID days. Everyone's wearing masks, which added another layer of complexity to the whole project. But it really required just tremendous team dedication to get it done. And as I mentioned, it was a large team, just to kind of hint at that. These are names of many of the folks involved in the first Tabula Sapiens project. So having done that, what did we do with the data? Well, let's talk about the second secret of life. This is Jacob and Monod, who discovered the so-called second secret of life, which is that some genes control other genes. That's also known as gene regulation. And we now understand how that works. What happens is proteins called transcription factors will bind to the genome and recruit other protein machinery to transcribe the gene. That's why you don't have every gene turned on in every cell in your body. There's repressors, which will turn the genes off, and other mechanisms that turn them off. And then there's proteins which turn them on that come and bind and signal, hey, let's turn this gene on and make RNA and transcribe it. And it's a very sort of complex system. And we decided to try to look at that in the cell atlas, and it came first in the fly. And so here is a diagram showing every column is a different cell type in the fly. Remember, we found like 250 of them. And every row here is a different transcription factor. And these 500 transcription factors represent about 2/3 of the total transcription factors in the organism. So these are the things, these are like very important genes because they turn on other genes. And many of them have unknown function. And what you sort of see is that many of them are only expressed in one or a handful of cell types. And so for the first time, we can tell the fly biologist where to go to study this particular transcription factor. Go to this cell type, that's where it's used, that's where it defines cell type, and you can work out its biology there. It's like a map to understanding how cell types are defined and which transcription factors are expressed where. And it was such a powerful tool that we decided to do the same thing on human. Now, if you look at the human genome, there's roughly 1,630 odd putative transcription factors. And these have been identified, some of them experimentally, but many only computationally. They're predictions. based on sequence similarity. So nobody knows what they do or if they are indeed really transcription factors. So we took that whole list and we said, can we see which of them are expressed in the cells we put in our Tabula Sapiens cell atlas? And it turned out every single one. So we've been able to find the cell types of origin of every single transcription factor in the human genome. And this is sort of what part of it looks like. Each row here, each column is a different transcription factor. Each row is a different cell type. We computed a statistic called tau, which is kind of specificity of expression. You can see that many of them are, like in the fly case, expressed only in a few cell types. And as we drill into well-studied cell types, we see ones that pop up that have been well-studied and known. And so we're recovering a lot of known biology here, which is great, but also discovering many new functions of transcription factors in the human body that have never been understood before. These are the ones that are very specific for different cell types. These are some that are very non-specific. You can see the difference. They're expressed in virtually every cell type. And let's talk about those for a moment. So here's sort of similar data. This is now in ascending order of that specificity statistic. And this was the 160 you saw before. This is the next group of also very ubiquitous ones. And so what comes out in this list of, let's say, the top 100 or so, are many well-studied transcription factors that are universally used. They're central to cell function, no matter what type of cell it is. Things like cell division, things like metabolism, things like stress response. Every cell has to deal with that. They're all using the same transcription factors to do that. And we're finding a bunch of them. But we're also finding a bunch of transcription factors that are predicted and have never been studied experimentally before. And so you can see those in a list like ZNF. Anything with ZNF is a zinc finger. They just got numbers. ZNF 121, ZNF 384, ZNF 639, so forth and so on. And so these are putatively transcription factors. They're used in every single cell, and yet biologists have never figured out what they're doing. And they must be very fundamental. And so this is like an amazing priority list for discovery of universal biology that people haven't run across yet. So we're very excited about the applications of that. So I hope I've helped convince you that these cell atlases are like very powerful phenotypic companions to the genome. That by providing a molecular definition of cell type, they help us interpret how the genome is used in different cells. It would be nice to be able to predict that computationally or theoretically, but to date, we've had to do it experimentally. Now, what I haven't talked about is sort of cell type identity as defined this way by transcriptome compared to the classical way of doing it was by physiology. And to make a long story short, we've done a lot of work in that respect and they line up very, very well. The two methods, there's a lot of concurrence. And so everything biologists have learned about cell type makes sense in this context. We're not throwing out the whole framework, we're augmenting it. And it sort of comes up that we're ignoring a lot of things about the cell. We're ignoring all the organelles and the compartments and the structure of the cell. And we're treating it sort of as a bag of RNA. I wrote an essay about that if you're interested in, what you get from doing that, what you give up, kind of the trade-offs. You get a lot. And it's useful for many questions that are very fundamental. But of course, it doesn't answer everything in cell biology. That's not the claim here. How am I doing on time, Sid? Okay, got time for another vignette here. So let me spend a little time talking about an application to human health of this approach. And this is this approach of doing single-cell transcriptomics and atlasing in the immune system. This is work that was done by a very talented graduate student in my lab named Derek Kroot, who's shown here, and in collaboration with Kari Nadow, who was running the Parker Food Allergy Clinic at Stanford before she moved to Harvard. We were interested in trying to understand the cells that cause food allergies. And they are cells that make a certain kind of antibody called IgE, which is very rare. Most people don't make a lot of that. And nobody had, in fact, ever purified those cells from humans in order to study their transcriptomes, and because they were so rare. And we worked out a way to do that using single-cell transcriptomics. And that's sort of outlined up here. We had blood samples from people in Kari's clinic. We would sort of enrich for B cells, but then do a lot of single-cell RNA-seq where we didn't try to purify out only the IgE-producing ones. We looked at all the different kinds of B cells and then went back afterwards, having done the informatics and figured out which ones were from IgE-producing cells, and then focus our analysis on those, both to look at their transcriptomes, but also to clone and produce the antibodies that were coded for in those cells. And so having the genes for the antibodies, we could make the antibodies and then test where they bind it. And lo and behold, many of them bound to the food allergen. So in this case, these were people allergic to peanut, and about half of the IgE sequences we cloned out and expressed would bind to peanut. And so we had found a way to kind of discover the antibodies that were causing the disease and to study them. And it's sort of interesting. Here you can see the relationships between those antibody sequences. So each symbol is a different antibody sequence that was cloned from a patient in the study. The patients are differentiated by different shapes of the symbols. The colors tell you something about the kind of antibody. IgE is the one we're interested in. And the size tells you about how mutated it was. And what you can see is there were some antibodies, like look at this CF1 up here. Okay, we're losing the battery. That's one antibody sequence, family of sequences that are closely related, where there's different patients had evolved the same antibody. So these are two different patients. Antibody creation is a process of evolution. Sequences are randomized, selection happens. The chance of two people evolving the same antibody is ridiculously small. And yet here were two patients who evolved almost exactly the same antibody that bound to peanut. And that's telling you something, that selection pressure is extremely high there. And that must be a very sort of potent antibody. And so we went and decided to analyze those antibodies and found that the sequences were highly mutated, which makes it even more exceptionally unlikely that they would have converged to the same sequence. And enriched in mutations that cause a change in the protein coding. And if you look at the distribution of mutations across all the antibodies we studied, these are at the way higher limit. These are some of the most mutated antibodies we found. Then we began to engineer them. So we started to change them around. We'd swap out different parts of the antibody sequence. We would unmutate the sequence, go to the naive sequence. We would swap with other sequences. And what we found was that we could destroy the binding by going back to the unmutated sequences. We could also tweak it a little bit. This is the naturally occurring one. So these are two different parts of the peanut, and the antibody bound not just to one part of the peanut, but two. And we could make it sort of trade off a little bit between the two regions. And that kind of protein engineering ends up being very useful for trying to develop a cure for food allergies. So Derek and I went off, we started a company together called IGenX. Derek is the CTO there. Kari is a founder as well. And they went and scaled this up on much larger numbers of individuals, much bigger studies. And here's kind of the same diagram showing now across thousands of antibodies, 3,000 antibody sequences across many different patients. Each color is a different patient, different subject. And you can see this convergent evolution is happening quite frequently for this type of antibody and people who have this sort of disease. And what we decided to do was engineer a different version of the antibody that would not cause allergic response. Okay, so you can keep the part that binds the peanut, cut off the part that drives all the inflammation, the anaphylaxis and all that, and administer that in principle to people, but certainly to mice, and see if that would soak up all the peanut and prevent the peanut from causing inflammation. And it turns out that that works pretty well in mice. So if you take a mouse model for peanut allergy and you administer this drug, all of a sudden, so down is bad. So if you take the allergic mice, if they're not allergic, they look like this. If they're allergic and you give them peanut, bad things happen to their temperature. They go down. As you give them the drug, increasing amounts of the drug, you reach a level where the mice don't have an allergic response anymore. So it's a brand new sort of therapeutic approach for food allergies, validated in mice, published a couple of years ago. And the company then launched a clinical trial in humans in Australia, which has just read out earlier this year, and it's working in humans as well. It's kind of amazing. So all this derived from the ability to do single-cell transcriptomics to discover those antibodies that cause the disease and then create engineered versions of them that would protect against the disease. So in the last couple of minutes, just to give you sort of heads up for some of what's coming tomorrow, I think especially in Toronto, which is World Center of AI, you appreciate that kind of amazing and somewhat scary revolution we're all living through with artificial intelligence. And these are pictures that I generated with DALL-E a while back. These are the prompts down here. I asked it for all the proteins in the bacteria or all the cell types of the human body in the style of impressionist painting. It comes with these amazing pictures, and it's really quite stunning. And so we began to think about ways to apply these techniques to understand the data in the cell atlas. Remember, 20,000 numbers for each cell. Millions of cells, up to hundreds of millions of cells now. And how do you assimilate all that and assemble it? And began to get interested in the idea of building large language models. To do that, when I was at CZI, we put together a sort of unified database to do that. Started at 30 million cells, grew to 100. It's going to be past 1 billion in the next year or two. So Yuri Leskovich and I and two talented students, Yusuf and Yane, began to explore this. The paper will be coming out shortly, and I'll talk in more detail about it tomorrow. We train a large language model, same way that ChatGPT is trained. You cover up some text, you cover up some of the numbers, try to predict them, and discovered amazing emergent behavior there. And then Theo Karaletos and I and the CZI team went on to build a next generation model, which is trained across huge evolutionary time, 1.5 billion years, and we're trying to use that to discover something about the origins of multicellular life. With that, I'll thank you. I'm happy to take any questions.
It's so good.
Clap around the mic, but if you're too far.
I can repeat the question. Yeah.
Yeah.
Being able to determine the structures of 1,000 or so proteins found in cells, and how that would help the research that you're doing.
Yeah. So the question is what I think about AlphaFold and its impact on science. And I mean, it's been amazing. It's been revolutionary. And, it's really changing the way people are doing basic science, the way drugs are being developed. It's hard to overstate its impact, actually. It's been terrific. And sort of related techniques are, the community is all over it, and results come out by the week. Sorry, up in the back.
And had that changed over time? Did you think you were going to do these kinds of big projects across huge teams?
Yeah. So the question is what I've learned from the large scale collaborations and the team science. And no, I wasn't really expecting to do that. But kind of the research evolved to a point where it was clear to get to the next stage of really doing the whole organism-wide measurement of an atlas, we would need a big team, and funding and collaboration and all that. And, it was tremendous fun to do. Everybody was super excited to be part of it because it was really on the cutting edge of science and their field. And we kind of took the Tom Sawyer approach in that, the thing where he painted the fence and said, it's so much fun, why don't you come do it? We didn't fund people to do the mouse cell atlas, actually. We said, we'll pay for the reagents, but you've got to supply the labor and the manpower and whatnot. And you'll get the first look at the data and you can write the paper with us. And everybody signed up to do that because they were so convinced they would discover, and they were right, that they would discover interesting things about the problems in biology they cared about. And so it was a great adventure. And, some fun serendipitous things came out of it as well, like the lemur. We weren't planning on doing lemur, which is a non-human primate. But our lung expert in the project, a guy named Mark Krasnow, was also interested in lemur biology. He had a lemur colony going at Stanford, and they had to euthanize a few of the lemurs because they were not healthy anymore. And the vets were saying, you have to, for ethical reasons, have to euthanize them. And he said, why don't we do a lemur atlas? We're going to sacrifice these animals. And we have the whole team fired up on mouse anyways. And so we just were able to pivot the whole thing. And in the middle of doing mouse, we're able to also do lemur. And that was kind of amazing. Yeah.
Wondering, technology side, what is kind of mistakes about producing these?
So the question is, what's missing on the technology side? Well, many things. I would say the technology has improved dramatically. When we were first doing this work, it was like $10 a cell. Now it's like 10, 20 cents a cell. So that has been continuous improvement. That's what's making it possible to scale up to billions of cells in the community without bankrupting everyone's budgets. So there's been continuous improvement on improving cost. What I've talked about here is mRNA, just the messages. Turns out there's most of the RNA is not in the cell, is not messenger RNA. There's all kinds of other species of RNA, and many of them regulate important aspects of the cell biology, so-called non-coding RNA. And the biochemistry has not really captured that to date. And so that's been an area of active research. And in fact, we just submitted a paper on a human cell atlas of non-coding RNA. And then there's the things beyond the RNA. The most obvious one is the proteins, but also the metabolites. And, I would say the single cell proteomic world has really turned a corner in the past few years, and it's finally really working reliably. And so you can see that that's going to scale up to answer a bunch of questions. And some questions you can answer with RNA, and some you cannot. And there's some that'll be better suited for proteins. And so that's now all starting to come online. Yeah.
Because they being a bit too brown, but how do these microfluidic cells actually separate out of this amount?
I'm sorry, say it again.
How, the cost of being too crowded with the house, how do these microfluidic cells actually separate out of the amount?
Okay. So the question is, how does microfluidics actually separate the RNA? They don't separate the RNA. So it turns out you can do this as a one-pot protocol. So you have all in the same pot, the lysis buffer, which breaks open the cell, the reverse transcriptase, which copies the RNA into cDNA, and the primers, which have the barcodes for the cell on it. So all that can be done in one pot, and then you can just amplify the whole thing. Yeah. I'm sorry? Topology, yeah. Mm-hmm. Yeah, so the question has to do with topology of DNA and how you map it back to sequence and things like that. So the topology happens on a length scale much longer than any individual sequence bit. And it's sort of known, there's a well-known topological problem in cell division. The genome is double-stranded, it kind of comes apart to be copied, but then you've got like a Mobius loop problem to make two daughter cells. And so there's enzymes called topoisomerases that will measure and change the topology. And that's all been very well studied. And they turn out, this is not esoteric. That ended up being, they turn out to be very important targets of cancer drugs, because cancer is about cell division. So if you can drug the topoisomerases, that helps treat cancer patients. And so that all happens at a level sort of beyond sequence. The part that is sort of sequence specific is not purely topology, but more geometry. People have been studying how the DNA arranges itself within the cell, even in bacteria, and it's not random. The genome is arranged in very stereotypical positions within the cell for reasons that are not totally understood yet. And that part is sequence connected because it's related to certain genes. Yeah.
Is this in the transcript going from the same tissues from different?
So you're asking if we found anything interesting, same cells, different humans? Yeah. Well, for sure. I mean, between health and disease, there's a huge set of things, and those are always tissue specific. I didn't get into it today, but both from us and many other people in the community studying things like tumors and how they're different than the normal tissue at the transcriptomic level has been incredibly informative, and has also led to the discovery of new drugs. The allergy one, for example, like you won't find those IgE cells in people who are not having food allergies or other kinds of allergies. Many things like that have popped out. There's age differences. We found that in the mouse, and there's ways to look at that in the human as well. Yeah.
You mentioned when you fuse older mouse, not fuse, but why an older mouse and the younger mouse?
Yeah, parabiosis. Yeah.
That the phenotype emerging or out the back.
Yeah. Yeah. So the question comes back to the parabiosis experiment. So what was learned about aging there? And that's something that the aging community has worked on for a number of years. And when you when the old mouse gets young blood, they definitely, by sort of number of gross physiological measurements, appear to be acting younger, behaving younger. And the molecular evidence we found in these studies sort of confirmed that. And we had an interesting opportunity to compare sort of natural aging, which we had done in the first study, with accelerated aging from parabiosis and rejuvenation, reversed aging, so you just change the sign. And we could ask, with the gene expression differences we saw in rejuvenation, consistent with what you see in natural aging, with the sign reversed, and with the accelerated aging, was that consistent with natural aging as well? Or were there artifacts from the parabiosis? I mean, it's very traumatic to do the procedure and all that. You can imagine, and I certainly was very suspicious there'd be a lot of artifacts because of the procedure. And it turns out that for the vast majority of genes that are downregulated with age, the parabiosis successfully reverses that, and it reproduces the opposite of what you see in natural aging. For the genes that were upregulated, those were more artifacts. So we were able to, with a fair degree of nuance, separate artifact from reality in those experiments. We don't think it's genome connected, phenotype connected. Yeah, yeah. It's not. I don't think it's about mutations. And there's a whole nother avenue about that. We could have a long talk. Yeah. Well, you're at one. You haven't had one. So the question is, how do you find these transcription factors that haven't been studied by sequence basis? Yeah. So it sort of comes back to this question about AlphaFold and protein shapes. And so, one of the underlying commonalities in biology is that, proteins that will bind the genome have often a very related shape. And the sequence that makes that shape are related. And so even though they may not have been studied, once you have all the genes in the genome and you can compare them to each other for sequence similarity, you can say, aha, there's a very good, it's a hypothesis, it's not a proof, but you can say the sequence of this unstudied transcription factor is reasonably close, and the domains that fold look sort of similar to ones of a well-studied transcription factor. You can then sort of make the hypothesis that the unknown one is a transcription factor, but you don't know it until you do the experiments. I'm sorry? Yeah, that's going to be a whole generation. Validated, then will be a whole generation of PhD theses, for sure. The zinc fingers, the structure is so well known, it's clear they're DNA binders. So there's very little doubt that they're actually doing something to the genome, but exactly what remains to be determined. Yeah.
Yeah. Getting analyzed cells and then the genomes being able to predict those based on like cell structure or cell function.
You're asking about what's the state of theory? It's sort of predicting cell biology. Yeah, well, you've got Sid here to take you through all that as a local expert. I think it's a fascinating frontier right now. And this thing about the central dogma was one specific question about information flow in the cell, about how information goes to protein synthesis. There's many other questions of information flow in the cell yet to be addressed and in the organism. I wrote an essay about that called The Cellular Dogma a couple of years ago to kind of lay out what I thought were some of the big open problems. And there's a bunch. There's a lot to do. Yeah.
I have the safety that it seems like your atlas studies kind of shift the question to, if I have the safety of how do I get RNA, big expression of RNA. Is there any indication of what the something that may do is with genetic? How do you get from, say, stem cell to say this bag of pattern?
Yeah, so the question has to do with how do you predict the cell types, the mRNA content from the genome? I think knowing the transcription factor function is going to have a lot to do with it. I think that's going to be a really important piece of it. I should say it's also not clear that it's possible to do it. I mean, it may be that you need more than the genome. I think formally you cannot rule that out. It may be that you need some of the proteins and metabolites that come along when the sperm and the egg meet to make the oocyte. That makes it even more interesting to me that you don't know that it's possible. My bias is I think there's probably a way to do reasonably well. on it. So I come down on the side of it's probably possible. But I think it's going to involve, really trying to digest everything we've learned from these atlases and finding ways to generalize it. And I think these large language models are going to be a big piece of it. We have one more? Yep.
The mouse was expected because of sort of additional knowledge associated in the analogy that you shared. And, because usually well, so was that surprising that, was that additional knowledge that went into, the information that made it expected it to be successful? I just wanted to know.
All right. So I think the question has to do with the expectation of success of the clinical trial. And, this touches on a sort of, common deep question in the drug development area, which is that at what point do you have enough confidence to try something in humans? And in this case, and the animals are not always predictive of what happens in humans. There are many examples of diseases cured in mice, but drugs don't work in humans. In this case, we were trying to prove a new method of action. This is not kind of a way that had been used to treat allergies, food allergies before. So having it work in mouse model was like an important, I say, brick in the foundation of our confidence to go into humans. But it's certainly, not, we're not 100% confident it's going to work. And in fact, it didn't work in 100% of humans. It worked in roughly half to 2/3. So you always learn things because human physiology is different than mouse. And so, we're now trying to figure out why it's working in half and not the other half, and getting to that point. But that's a not uncommon thing in drug development.
We have time for a couple more questions if you're up for it.
Sure.
All right, so we'll take last 2 questions if there's still more. Okay, the back, I guess, yeah.
Yeah. So the question has to do with the beautiful broccoli diagrams, and my friend calls them broccoli. And what does it mean? And do they always separate and differentiate? And I'll put one back up so you can see what I'm talking about here. Give me 1 second and I'll find one. Let's see. All right. Like this, right? And so, first of all, let me just say a little bit about it, okay? This is not a linear projection by PCA or something like that. These are highly nonlinear algorithms like UMAP and things like that, that are doing things that try to preserve distances. Things that are close in the high dimensional space are close in this space, but not necessarily when you get to further, longer distances. So it's complicated, it's tough to interpret from these things. You got to be careful about conclusions you draw. They're more for visualization. But it's also the case that I think what you're getting at about sort of continuous variation of cell types, it's wrong to assume that all cell types are discrete. That's another simplification that often happens in the field, that you sort of impose upon your data this notion that it must be decomposable into discrete cell types. So we found a number of examples where the variation is continuous. Often it happens in the brain, actually. And you do see that there can be continuous variation from one cell type to another. It's always important to be mindful that that is one of the possibilities, and biologically it happens a fair bit.
Take one last question.
Actually, it's also, so I'm very curious, Steve, that within each larger than the separation cell types. Is that significant, or is it just hard to back up?
Yeah, so the question has to do with the kind of size of the cluster versus the distance between them. And it's artifact. There's a lot of measurement noise here. And most of the variation within a cluster is measurement noise and not biological at this point. And also you have the whole distortion of the mapping.
Okay. I guess with that, let's thank Steve one more time for this exciting talk.